Hello friends, in the last class we had studied about the non-cyclic photophosphorylation. Today we will be studying about the next phosphorylation process, which is the cyclic photophosphorylation. Now, as I told you in the last class also, photophosphorylation is a process by which ATP synthesis will take place in the presence of light. So let us again start. There will be a reaction center with chlorophyll A molecules there. These are all the accessory pigments or proteins associated with the chlorophyll A molecule, which are also going to trap light and ultimately transport it to the chlorophyll A reaction center. So let us suppose that when the sunlight falls on the chlorophyll A molecule. Now, in case of cyclic photophosphorylation, the type of photosystem that is involved is PS1. And you already know that PS1 is that photosystem which generally absorbs light of 700 nanometer. So, when the sunlight is falling on chlorophyll A, it is going to absorb the 700 nanometer of light. All the other accessory proteins or the pigments associated with the antenna part, they are also going to trap light of different wavelengths, and ultimately all but that wavelength will be passed on to the chlorophyll A molecule. And as a result of these process, what will happen is a pair of electrons will get excited. And these excited electron pair now will be transported to the primary electron acceptor. This primary electron acceptor is then going to pass these pair of electrons to the different electron transporters. Now, here in this case, like we had studied in non-cyclic photophosphorylation, there is a complete electron transport system involved. Similarly, here also there are different electron transporters involved in the electron transport system. The first pair of electron will be actually accepted by a molecule called as ferredoxin. The ferredoxin is then going to pass the one pair of electron to PQ, that is the plastoquinone. Plastoquinone will transfer the one pair of electron to the next electron acceptor, which is the cytochrome complex. And ultimately, this cytochrome complex is going to transfer the two pair of electrons to the last electron acceptor, which is PC, or you can say plastocyanin. Now, when this plastocyanin is going to receive the electron, after this, what happens is these two pair of electrons will be again transferred back to the reaction center having chlorophyll A molecule. So what happens here is, once the electrons get excited from the reaction center, they pass through the electron transport system. So the energy, when they are transferred from one transporter to the another, and the energy will keep on reducing. At last, plastocyanin is again going to return back the two electrons to the reaction center. And hence, this photophosphorylation is called as cyclic photophosphorylation. The electrons that get excited from the reaction center are once again returned back to the reaction center only. And when these electron pairs are actually transported, when one electron pair is transported from ferredoxin to plastoquinone, this energy that is released by the two electrons will be used to form one ATP molecule. So this is the ATP that has formed here. Again, these two electrons, when they are transferred from the cytochrome complex to plastocyanin, reduction in the energy of the electron is again helping for the formation of ATP molecule. So what you can say here is during cyclic photophosphorylation, there will be two ATP molecules which are formed, and there is actually no reducing power involved or no NADPH2 formed. So this photophosphorylation process is called as cyclic photophosphorylation. If you see the characters or how does it differ from the non-cyclic photophosphorylation, what are the different things that you can consider? The first point that you need to remember is the cyclic photophosphorylation involves PS1 only. There is no PS2 present in cyclic photophosphorylation. The second thing is that 2 ATP will be synthesized, right? Now, the electrons which are finally released by the cluster sign, since they are returning back to chlorophyll A molecule, there is no external source required for the transfer of electron to the chlorophyll A molecule. So, there will be no splitting of water molecule taking place in cyclic photophosphorylation. Then, no reducing power is involved. When I say no reducing power is involved, what I mean to say is there is no formation of NADPH2 taking place in the cyclic photophosphorylation process. Now, since NADPH2 for the dark reaction, that is the biosynthetic phase, NADPH2 and ATP both are necessary. And since cyclic photophosphorylation is only involved in synthesis of ATP, the more favored and the more common form of photophosphorylation which takes place in plants is non-cyclic photophosphorylation. Cyclic photophosphorylation is conditional, okay, as it is going to absorb 700 nanometer of light and only ATP will be synthesized. It is conditional. Where exactly does this take place? Location. Like we had studied for the non-cyclic photophosphorylation, there is the formation of ATP is taking place towards the inner side in the thylakoid lumen, and the NADH2 is synthesized on the stromal region. Both are present, right? In case, or you can say that the non-cyclic photophosphorylation system is present in the thylakoid membrane. So you can say that the thylakoid membrane has both PS2 and PS1. Now, these cyclic photophosphorylation, basically you can say it takes place in the stromal lamellae. So, if I draw a diagram here, this is a thylakoid membrane, double membrane structure, which is attached to a stromal lamellae, and what you can say is the thylakoid membrane has both PS2 as well as PS1, whereas the stromal lamellae is going to have only PS1 system. This is PS2, PS1, right? So there is no NADP reductase enzyme associated with the stromal lamellae. So there can only be cyclic photophosphorylation which takes place, whereas here the non-cyclic photophosphorylation takes place. Okay, so this is about the two types of your photophosphorylation reactions. In the last class we had studied about non-cyclic, and this is the cyclic photophosphorylation. Thank you.